So same method. So what is this method? What is this method known as? This method is called as microarray analysis. This method is called as microarray analysis. Microarray. Still my question is not completed. Microarray based, this is the one procedure, microarray. In this microarray analysis, what we do? Take the sample of the patient, go to the gene library, there will be one chromosome which will be having entire human genome, and we have to hybridize them. And now we have to see that is there any defect in this hybridization or they are complementary to each other. So if there is a defect, it is a sign of tumor gene. Fine. So microarray analysis, what I said you, a hybridization between two chromosomes. So by this hybridization, what we are doing? What we are doing? Comparing. So this method is called as microarray based comparative genomic hybridization. So this methodology is called as microarray based comparative genomic hybridization. Comparative genomic hybridization, and this was the option in AIIMS exam. Shortly they have written CGH. So this was the answer in AIIMS exam, November 2012. Same chromosome, tumor and normal gene, both we have to detect. Best method will be comparative genomic hybridization. So this will be the best method, comparative genomic hybridization. And this is called as microarray or array based. Sometimes they write like this also, array based. In Robbins it is written like this only, array based comparative genomic hybridization. I wrote like this for your concept and understanding. For another question, this was All India question, microarray analysis. This was All India 2013 question. Microarray analysis, what is the basic principle? What is the basic principle of microarray analysis? What is the basic principle of microarray analysis? So now you can see this microarray based comparative genomic hybridization. Basic principle. So name itself is telling basic principle. We are taking two gene, normal, our patient, and gene library gene, and what we are allowing? Hybridization. So this is the principle. Hybridization is the principle. So principle is hybridization. So this was right answer. Microarray analysis, principle is hybridization. This is one question. All India NEET 2013, there was another question. The microarray analysis, what is their use? Option 1, gene expression profiling. Option 2 was molecular classification of the cancers. C was both of the above, none of the above. So what will be your right answer? Microarray analysis is used for gene expression profiling. So number 1, microarray analysis uses, I am writing here, uses are gene expression profiling. Gene expression profiling. What is this gene expression profiling? What is this gene expression profiling? We are making separate groups according to basis of the genotypic variations. For example, if you take example of breast cancer, we classify their cancer like this, which are cancers estrogen receptor positive, some cancers are progesterone receptor positive, some cancers are HER2 new positive. So all these are important genes, ER, PR, HER2 new. What is this? Gene expression. So we are separating all these genes. So this is called gene expression profiling. All genes we are making separate making profile for ER, PR and HER2/neu. So this is the basis for what? What is this? This is basis for what? Molecular classification of the cancer. Which cancer? Breast cancer. So this is another use of microarray analysis. So microarray analysis we can use for gene expression profiling, and we can use it for, now you can see here, so this is a molecular classification of the cancer. Molecular classification of cancers. So CA breast, and now if you see your new Robbins, you will find that CA breast have been classified, molecular classification into 5 groups: luminal A, luminal B, basal-like, normal cell-like and HER2/neu positive. And so these are 5 groups of molecular classification of CA breast. So just remember, what is role of microarray? To separate gene expression profiles. Why we are separating gene expression profile? To classify cancers on the molecular basis. So these are 2 functions. So you can see, so answer will be both of the above. What is use of microarray analysis? Both gene expression profiling and molecular classification of the cancer. Principle